Good evening, everyone. Thank you for joining in for the live session. Before we start with our session two, I would like to know if you have any doubts from the previous session or any clarifications. I'll just give you 2 minutes. So, if you have anything, you can either ask by unmuting yourself or putting it in the chat box. And before that, I guess my audio is clear and also the chat box settings is working now. Fine. Okay, thank you for the responses. So now I'll start share my screen and we'll get started with the session. Okay. I guess the screen is visible. Okay. So yes, welcome to the second live session for the course Applied Environmental Microbiology. I am Guru Priya, Prime Minister's Research Fellow in the Department of Civil Engineering, IIT Madras. So I'll be handling all the live sessions for this particular course. So for those of you who have joined newly, um, I would like to tell that this will be kind of a practice session. So I'll be giving questions first, which I'll be asking for answers, and then we'll be discussing on all those questions. Okay, so you can ask your doubts at any point of time by putting it in the chat box. I'll be happy to clarify those. So before starting in with today's session, we'll see what are the course content that was covered in week two. So it was genetic code and translation was covered. We saw about the central dogma in last week's session also. So in continuation with that, there is a content about genetic code and translation, amino acids and protein structure, and then we move on to this part of bacterial growth. So we saw there was lectures about growth curve, microbial cell counting, and then coming to energetics, there is lectures about energy storage reactions and what are the nutrients that are required for microbial growth. And finally, there was also a lecture about fermentation and respiration. So having that said, now let me put on the first question for the day. Okay, so you can take your time in reading the questions, the statements, and giving your answers. Okay. And to any of you who is new to the session or to microbiology itself, you need not worry. We are just going to discuss all these questions and the concepts behind them. Okay, so I got quite number of responses now. So alpha helices and beta strands form the tertiary structure in protein. So all of you have almost answered that as a false statement. So if if not for tertiary structure, then what do alpha helices and beta strands belong to? Right. So yes, your answers are right. Um, we all know that all the enzymes are made up of amino acids, and they are mostly 3D structures. And also that any given protein will have a primary, secondary, tertiary, and quaternary structure that will give it distinct properties. We'll discuss it during that discussion session. And enzymes are biocatalysts. Yeah, they are biocatalysts. They are going to catalyze biochemical reactions, and they are mostly proteins. Yes, only few enzymes are there which are RNA enzymes. So otherwise, mostly enzymes are proteins. And you also have to be careful. As I told, few enzymes are RNA enzymes are also present. So it is not always that all enzymes are proteins. Okay. Here is a second question. You can just think of how the structure, what is the composition of the amino acid, and then you can answer. Okay, all of you answered B, and B is the right answer. So while learning about amino acids, one easy way to learn about amino acids is to remember what are their properties, how are they classified. So based on that, yeah, aspartate is not a positively charged amino acid but a negatively charged amino acid. As we all know, it is aspartic acid. So acids are going to contain COOH group, and it's going to be COO minus charge on them, and they're going to be negatively charged amino acids. Again, you can try answering the third question. We'll soon move on to the discussion for this topic. Okay, I've started getting responses for this question now. Right. So here D is the right answer. We all know start codon codes for methionine because methionine is the first amino acid in any protein sequence. So option C, all of you would have probably known it's true. Okay, and stop codon is a nonsense codon because it doesn't code for any amino acid. And in the genetic code table, if you see, you can see tryptophan being coded by only one codon. So regarding this Vogel base pairing, um, this statement is false because Vogel base pairing will decrease the effect of DNA mutations. So with an image, we will see why here. Okay, starting with the discussion regarding this amino acids and protein structure. First of all, we can see this genetic code here and the amino acids here. We all know proteins are made up of amino acids. Okay, so the primary structure of the proteins is the arrangement of the amino acids in the polypeptide chain that constitutes the primary structure. We can, as you can see here, maybe I'll just zoom in for this. Okay, I guess it's much clear now. As you can see, there are many different amino acids and they are classified uh, based on their structure and their composition. So regarding that question that was asked, you can see tryptophan has ring structures, aromatic structures. Similarly, the other two amino acids that contain this aromatic structures are tyrosine and phenylalanine. You can see valine, leucine being simple amino acids. Glycine, you can see a simple structure over here. And there are two acidic amino acids which are aspartic acid and glutamic acid, which we can refer to as aspartate and glutamate. So they are mostly negatively charged. And you can see amine group containing amino acids, asparagine and glutamine. So these contain amine group. So there are many differences in their composition, their bonding and their structure. And these amino acids are like linked together with peptide bond. As you can see here, this is the peptide bond. You can see this is one amino acid. So this one part is basically the amino acid structure that is represented in common. You can see an amino acid will contain a central carbon atom, will contain one COOH group and one amine group here and a side chain. So we call it amino acid because it contains one amine group as well as one acid group. So that is why it is termed amino acid. And this R can be replaced by many different things. So by replacing this R with different chemical groups, you get all these amino acid structures here. Okay, so when two amino acids are going to bond together, so for a polypeptide chain, you need this bonding between amino acids, right? So when two amino acids are going to bond together, you can see the OH group from the COOH of one amino acid and the H from the amine group of the next amino acid are going to condense to give away water and form this particular bonding, that is the C double bond O NH bonding. This bonding is known as peptide bonding. Okay, so this forms the basis of proteins. Without this bond, we wouldn't have had anything called proteins here. Okay, so these amino acids, where do they come from? We all know, as we have learned in the central dogma, there is DNA which transcribe to mRNA, right? So these mRNA, what do? They undergo translation. So during translation, we have learnt about triplet codon concept, where there there will be reading of three codons, and the three codons together will code for one amino acid and the tRNA. So this structure present in the bottom of the slide, this is the tRNA structure. So this tRNA will bring in the amino acid that corresponds to the triplet codon in the mRNA strand. Okay, so you can see the genetic code table here. We all know mRNA will be made up of nucleotides, right? So the nucleotides can have either they can have uracil, cytosine, adenine, and guanine. Okay, so you can have a, you must have a triplet codon. So you can have either U or U and C. Similarly, you can have all these combinations, and you can see, uh, for example, if you take phenylalanine can be coded by either UUU, UUC, or UUA, which means that when your mRNA is going to have either of these three codes in that place, the corresponding amino acid that is going to fill up that position in the protein is going to be phenylalanine. Similarly, when you go for lysine, you can see you have AAA and AAG. Okay, so here you can see that our AUG, which we know is the start codon, corresponds to methionine. Okay, and you can see that methionine is also coded by only uh, one amino acid. Similarly, tryptophan is also coded by only one amino acid. So in the question that was given previously, if it's either tryptophan or methionine, whichever is asked, they are coded by only one amino acid, whereas that's not the case for other amino acids. So one triplet code will code for a particular amino acid only, whereas the vice versa case is not there. Like glycine can be coded by either of the four amino acids. Okay, so this concept you have to have in mind. Having said that, then okay. Yes, Darshini, do you have a doubt? Darshini, you can either unmute or you can put it in the chat box.
Nothing, ma'am.
Okay, okay. So yes, now we have a strand of mRNA which will be having nucleotide sequence. So then our ribosomes will come bind to it. Our translation process will start when this AUG triplet codon is found, and as the thing proceeds, what happens is the tRNA is going to have an anticodon. For example, the mRNA sequence here has a GGU, okay, and this has a CCG thing, okay. So they are complementary, so they will bind, and this GGU is going to code for glycine. So that amino acid will be transferred here by this tRNA. Now you can have a doubt that how can a G pair with U? This concept is called as vowel base pairing, where if the first two are perfectly matched, the third one need not match necessarily. So any of you have a guess why verbal based pairing is beneficial, or why is it occurring? Any of you, can you yeah, there is something in the chat box. Okay, so this is because if you have to have you can see these many triplet codons are present in the genetic code, and when you have to have tRNA that contains the anticodon of these many genetic codes, it's going to be biochemically more energy consuming for the cell. There will be more load on the cell metabolically. So in order to reduce that, this verbal base pairing concepts usually come into picture. So here you can see the glycine mRNA codons are GGU, GGC, GGA and GGG. Okay, so but the tRNA anticodons can be CCG, CCU and CCC only. For these four, you will have only you can have three anticodons, which will reduce the metabolic load of the cell in producing. You need not have numerous tRNAs in the cell for the translation process to occur. So that is why this verbal base pairing is beneficial. So in our question, we saw that regarding mutation. Um, in the case of mutation, what happens is maybe one of the nucleotides is removed and some other nucleotide comes in that place. So if it's going to be a very hard and fast thing process here, then the mutation will cause some other amino acid to bind. So the entire structure, the function of the protein may even get distorted in that case. So this adverse effect is being reduced by this verbal base pairing concept. Okay, so that is why as I mentioned in the question, it was not amplifying the genetic mutation effect, but instead it was this verbal base pairing decreases the mutation effect in the organism. So now we know that we have genetic code, we have amino acids. These codes bring in the correspond to amino acid which are brought in, and finally at the end of translation process, so like this primary structure, we get a chain of amino acids that are linked together by a peptide bond. Okay, uh, these peptide bonds in turn, they can take alpha helical structure or beta pleated structure. You can see the difference here. Here you can see it is in a helical form. Okay, the bonding, the intramolecular forces will be making it in the form of a helix. Okay, so this is the alpha helical form, and the another secondary structure is the beta pleated sheet. You can see that they are arranged in the form of sheets here. Okay, so from this primary structure, due to intra bonds, you can get this secondary structure. Now the important thing is uh, these intra bonds happen due to the presence of these different kinds of amino acids. You can see you have positively charged amino acid, negatively charged amino acids. Basically you know that this positive and negative charges have a attraction between them. You can see electrostatic attraction, or when there are functional groups that can form any other hydrogen bond or van der Waals force. So because of all these forces, you can see structures like this forming. um, Again, because of strong forces, you can get the tertiary structure also. Okay, the difference here is this forms a three-dimensional space. So as you all know, enzymes are proteins, right? So in the case of enzymes, if you see, um, in the case of enzymes, if you see, you have an enzyme like this. You will have some space here. Okay, I can't draw a three-dimensional image, by the way. So you can see a space here where some substrate can come and bind to. So here you need to have some space for the binding, and you also know that the enzymes are very specific, which means that only the target substrate which has to be catalyzed can come and bind to the enzyme. So their arrangement, why is it specific? Because their arrangement is made up of such amino acids which forms a specific site here called the active site, where your substrate can come and bind. So why I am telling this is a three-dimensional structure or a quaternary structure in which two tertiary structures uh, can be bonded together. All these confer this specificity in the case of proteins. You know, there are many. I gave one example of this enzyme. You know, in our plasma membrane, like in our plasma membrane, there are some transporter proteins here. So they need to have some specific shape and structure, and that will also define their function. So that is why, for example, there are some molecules here, they are transported from this side to this side. In case of facilitated diffusion and all, you see some mechanisms where only specific uh, molecules can be bound to some specific um, transporters, okay, and they'll be either taken into the cell or taken out of the cell. So in all these cases, proteins are involved. Proteins almost are involved in all the processes in our metabolic processes, be it prokaryotes or eukaryotes, okay. So therefore, all these functions are possible only because they have specific structures to it, and these structures is brought about by their uh, amino acid composition and the bonding between them. So that is the significance of this primary structure, secondary, tertiary, and quaternary structure. Okay, I guess this concept is clear for you. If you have any doubts or if you want me to repeat any of it, you can let me know. Can I move on to the next question? Okay. So this is the next question. So now we will be moving on to microbial growth, and you can answer this question. Okay. So yes, I I hope you all are clear with what logistic growth model means as it was explained in the lecture. So we all know nutrient limitation plays a very important role in the logistic growth, because once the nutrient gets limited or there is accumulation of toxic materials, there is a stationary phase coming in. Now you can try answering this question. I'm sorry, this is direct methods for microbial cell measurement in the option B. Okay, now we have a set of varied options in the answers. Um, I'll give you 1 or 2 more minutes to tell me what is wrong in the answer that you have told. Like what is true, or why are the other options false? Anything, any explanation? Because we have got B, D, and C in the answers, I guess, from you people. Okay, are you confused now? Seeing varied responses, let's discuss the answer only now. So, option C is the right thing. I have told you to identify the true statement. So, turbidity. Uh, if you have, okay, I got a response. We'll discuss regarding the B option also now. So now regarding this turbidity, if you have performed some microbial growth experiments in the lab, like I don't know what background you people are from biotechnology, microbiology, or something related to it, or something different, completely different, being civil engineers, chemical engineers, or something. So when you're going to perform microbial growth curve experiment, or generally a microbial growth in a conical flask, you will first prepare one medium. Medium means nothing but the nutrients for the growth of the microbes. Okay, so when you are going to prepare the medium, you will again keep it in heat, and you will do a procedure called autoclaving. So that procedure will uh, kill all the other microbes present in the media before you going to inoculate your microorganism. Okay, so during that time, the media and the conical flasks will be very clear. Okay, as you are going to inoculate, inoculation means adding your microorganism into that conical flask. So after some growth, the turbidity is going to increase. So when the turbidity is very low, it doesn't mean signifies exponential phase. No, during exponential phase, the turbidity is going to increase, and it will be higher than previous. So that statement is not true. Measurement of turbidity and plate count are direct methods for microbial cell measurement. I guess this statement is a bit confusing here. Let me explain what is direct methods for microbial cell measurement. In direct method, you're going to directly count. Okay, see, if I'm going to handle this session in person with you all, if I'm going to count how many of you are present there, that is called direct method. Or if I'm going to just have some other metrics which I can equate to the number of people that are available in the room, that is known as an indirect method in terms of microbial cell measurement. So you know that microbes have very fast reproduction, so they double very fast and they're going to grow very fast. So counting individual cells is a bit of a tedious process. That is a direct method. Plate count is obviously a direct method, where you are going to do that streak plate technique that was taught in the lecture, and then see how much colonies your sample might contain based on your dilution and stuff. Okay, but in turbidity, what you're doing is you're going to measure only the optical density, right? You're going to do OD measurement. So that is not a direct method, but that is an indirect method. So this statement is not true because I included the word turbidity here. If it's going to be only plate count, the statement is true. Okay, coming to this statement, the extent of lag phase depends on age of the inoculum. Uh, I guess now by now you will all be knowing that we have lag phase, log phase, stationary phase, and the decline phase. Okay, the lag phase is the first phase where the microbe gets adapted to the situation, or the nutrient condition or the environment there. So it might be feeding on some other sugar, you might be taking and putting it in a medium which contains some other sugar. So it has to prepare itself for surviving in the environment that you have kept the microbe in. Okay, so that is known as the lag phase. I think this concept was somewhat explained in the lecture that when you're going to put a microbe from exponential phase, like I'm going to have one um, already a media containing a microbe, okay, and I'm going to transfer it to another fresh media. So this inoculation process can happen with any age of the microbe that I had previously. I can put it from either the exponential phase or stationary phase, depending on from what phase I'm putting the microbe into the other fresh medium. The extent of the lag phase is going to vary. Usually what we do, we take mid-log phase microbes and then inoculate them into a fresh media, because they will have a fast, they can adapt themselves faster and they can grow faster. But you're going to put a stationary phase bacteria, it is already tired of growing. Okay, so it has to revive itself back uh, to just grow or multiply in a new medium. So that will take time. I guess you understand what I'm explaining here. So based on from what age of the inoculum you are going to inoculate, the extent of lag phase will definitely vary. Okay, so that is why it is a true statement. Coming here, when the microbial population is lesser than the carrying capacity, microbial death starts to occur. I guess you remember a graph where we have the carrying capacity K in the microbial growth kinetics that we have studied. So we have K. So when you have your N naught less than K, obviously growth is going to occur, and until K is reached, right, in the graph. Okay, I don't know if you are able to visualize it. Let me draw that only. Okay, so you have your K here, but your N naught is here. This specifies the number of cells. So this axis is the number of cells and this with the time. So when you have N naught less, what happens is it goes here until the carrying capacity is reached. But when your N naught is greater than K, what happens is it tends to come to K. When the microbial population is lesser than the carrying capacity, growth starts to occur, whereas when the microbial population is greater than the carrying capacity, death starts to occur. Okay, so that is the difference here. I hope this is clear. So is this clear? Because you gave varied answers here. Are you all convinced that C is the right answer? Okay, I see few thumbs up here. Okay, and responses. Okay, now you can answer this question. I guess this is an easy question. Right. So when we were discussing about this microbial growth, if we are not going to take the decay term into consideration, then we mean that uh, the the microbes are going to multiply infinitely, and that is not logically right. So incorporating decay term is very important because it incorporates the removal of microorganisms due to natural death, obviously due to various reasons, because of nutrient depletion, because of toxic accumulation, anything might be the reason. So decay term is very important in the case of growth model. So now we will see this. So this is what I was telling you, the bacterial growth curve. This is called the bacterial growth curve, where you plot the log number of cells you identify versus the time. Okay. So the first phase in any microbial growth is a lag phase, as we have discussed. It is the phase of acclimatization. It gets used to the nutrients and the environment, and once it is used to the situation, you can see logarithmic growth. So they tend to multiply very fast, they tend to utilize the medium that you have given, and they go into this log phase. And then there comes the stationary phase. This stationary phase may be due to nutrient depletion or toxic accumulation. So here is the stationary phase, and once they are not able to survive there in the medium that you have given, they then go to the death phase, where there is again a almost logarithmic decrease in the number of cells. So any guess why this stationary phase is applied to here? Why not this directly going down here? This, why is there a stationary phase? What it signifies? Right, that's the term. The net growth is zero here. Here you can see you will get a positive slope. Here you can see you will get a negative slope, speaking mathematically or graphically. But here you assume that the growth rate and the death rate are the same. So there is no net growth here. Okay, the net growth is zero. I guess this growth curve is clear. If you have any doubts in the growth curve, you can just put it in the chat box. Now coming to this microbial cell counting part, as I told, there are two methods, direct and indirect. So there are two direct methods, that is plate count and cell counter method. What I do is, like, I think you all know about the grid with which I think in the laboratory we usually do red blood cell countings, right? So in that you can see there are Petrophosphor grids present, with which you count the number of cells. Similarly, you can count, say, as the way you do with RBCs, you can count microbial cells also in that. Okay, so that is known as a cell counter, where you are directly going to see the cells present and count 1, 2, 3 in that. So a cell counter and a plate count, where you do serial dilution first and then plate them using a streak plate technique and see how many individual colonies are present. So in that way you can quantify, like, okay, after this dilution in this ml, this many microbes are present. So in my initial sample, how many microorganisms will be present? So using that simple concept, when you are going to count the cell, it's going to be a plate count method or a cell counter method, in both of which you are going to see the cells and count them directly. Okay, so that is what is known as plate count or cell counter. Both fall in the direct counting category, but when you come to the indirect measurement, not even cell counting, indirect measurement, you can see you will have turbidity, metabolic activity, and dryness. Any of you have any questions? If you are having questions, please mute yourself.
मैं थोड़ी देर में रुकी हूँ कोई चीज चीज
Okay. Okay. Sorry for the disturbance. And yeah, as I was telling, there are three more methods like turbidity, metabolic activity, and dry weight. So in turbidity, as I told you, you will be measuring using a spectrophotometer the optical density, and you will equate. Okay, if this is going to be my OD, then these many cells may be present. So that will be an indirect measure again. If you're going to measure metabolic activity, you will focus on any specific activity, perform the test for it, and equate it with the number of cells that might be present or the concentration of cells. In dry weight, as you all know, you are going to take your culture, dry it in an oven, and you're going to get the weight of the cell. Stoichiometrically, you can relate. Okay, if this is, if you know the composition, you can relate. If this is going to be the dry weight, and based on the individual composition and all, what will be the number of cells that will be there. So all these methods are estimation methods. They are not going to tell you exactly how many number of cells are going to be present with more accuracy. So that's the difference between direct and indirect method. I guess the amino acid protein part and this microbial growth part is clear for you now. So here is a very simple problem which you can try, and I'll give you a few minutes and then we'll discuss how to go about with this. Maybe I'll read the problem for you once. If one starts with 10 to the power 4 cells in a culture with a generation time of 2 hours, how many cells will be in the culture after 48 hours? So I give you 10 to the power 4 cells, and I tell you that the generation time of the microbe that I have given to you is 2 hours, and I ask you to grow it for 48 hours, and I'm asking you to report at the end of 48 hours how many cells you will be having. I hope the question is clear. You can try solving this problem for a couple of minutes and then we'll discuss. Any of you who have no idea how to start about with this problem? Okay, I guess all of you are trying, so I'll give you some time. Okay, I have got one answer. I guess the rest of the others are trying, so I'll wait for another couple of minutes. Okay, are others able to try this question? Can I wait for your answer for a few more minutes, or can I show the solution for this problem? Okay, okay, so here is the solution for this problem. When you have to solve any problem with a generation time that is being given, you have to use this relation where um, the number of cells that are present after n generations will be equal to 2 power n into the initial number of cells that are present. So the answer is very simple. In order to calculate the number of generations, what you will do is you will calculate how many generations will be covered in this time that I have given you. Okay, so 48 hours, and one generation will take 2 hours to grow, right? To get one generation, it will take 2 hours. So 48 hours by 2 hours will give you 24 generations. And I initially gave you 10 to the power 4 cells. So it's going to be 2 to the power 24 into 10 to the power 4. That will give you 1.7 into 10 to the power 11 cells. As Nidhi Praveen has answered rightly, it is 1.67 or 1.7 into 10 to the power 11 cells. Is this clear, or like you have any doubt? I guess it's a very simple problem. So if you have any doubt, or you can ask me, or can we move on to the next? You can give a thumbs up if we can move on. Don't see any reactions here. Okay, right. Thank you. Thank you for the response. Okay, another a formula related question here. I guess this is pretty much easy for all of you. You can try answering this question. You can find the right equation with regards with the change in Gibbs free energy. Okay, I've got two responses now. I'll wait for a few more to respond. Okay, there are a handful of responses now, so we'll discuss. So we all know for any system, if you are going to calculate the Gibbs free energy, you have to take into consideration the standard Gibbs free energy, and then about the constant or the K factor. Okay, so here you can see many confusing options, but you all of you have chosen del G is equal to del G naught plus RT ln K. That's obviously the right answer. And here uh, A is basically completely wrong. It's not negative. You're not going to subtract anything from the standard Gibbs free energy. And C, if it's going to be 2.303 RT log K, and then this is going to be right. Okay, the relationship between ln and log you all know. So if it's going to be 2.303 RT log K, then this relationship holds true and meaningful. Um, anyone have any idea if this is uh, what's the mistake in this D? So you all know the standard Gibbs free energy, right? Okay, no issues. If it's going to be um, for del G naught, then you should be having a minus sign here. Okay, so for any system, the del G naught will be when you are talking with a chemical system. Uh, in some cases, this can also hold true, but when it's going to have a positive sign, it will not have any meaning here. So the right option is del G is equal to del G naught plus RT ln K. Now you can answer this question. So now you know that we have completed about uh, microbial growth curve and growth. So now we are eventually moving into the energetics part. Okay, there is one response. Okay, many of you have answered A, and A is the right answer. So most energy-rich compounds in cell primarily are phosphorylated compounds with ester and anhydride bonds. Uh, Something that confers this energy to the compounds is the phosphate bond. As we all know, ATP is the energy currency. That is phosphorylated with three phosphates. So this is adenosine triphosphate. So phosphate usually phosphate bonds usually are made with the expenditure of energy, and when they are broken down, energy is released. So that is why the compounds are named as energy rich compounds. Okay, another easy question for you for the day. I guess this is a pretty straight question, right? Are you waiting for some twists and turns in the questions? Okay, a mix of A and B now. Okay, there are enough number of responses, and now, okay, I'll wait for another 1 minute maybe. Now tell me, there is an equal mix of A and B in the answers. Why A or why B? What made you think it's A or what made you think it's B? You can just type in 2 or 3 words, like not even a proper formation of sentence is required. Okay, okay. Anyone who answered A would like to add on any comments? Why that was A? Okay, so we have two reasons. See, the answer is aerobic oxidation because in anaerobic oxidation, ultimately, okay, we have one more explanation here. Thank you for the response. So in anaerobic oxidation, what generally happens is the energy is not generally available for growth and replication, whereas products are formed there. For example, if you take in the case of anaerobic digestion, if you take, you can see that if you are going generally we do in environmental remediation we use microorganisms, right? That is what we are studying about, applied environmental microbiology. Okay, so there are two processes where you can have an aerobic process, for example, activated sludge process and an anaerobic digestion process. In both these processes, if you compare, this aerobic oxidation will give us more biomass at the end of the process. More biomass in the sense more cells. Okay, whereas in anaerobic oxidation, there will not be more biomass present after the remediation in the sludge. Okay, so this is because in aerobic oxidation, with the use of the energy available, growth and replication happens, whereas in anaerobic oxidation you get compounds like the fatty acids, acetate, methane, which is ultimately the product of anaerobic digestion. Okay, so the kind of reaction that has more energy available for growth and replication is obviously aerobic oxidation. So in order to have this concept right, remember this example of this ASP and anaerobic digestion, the activated sludge process and the anaerobic digestion part. Okay, um, right. Here is the next question for you. Nidhi Praveen, can you unmute and ask your question uh, so that it'll be clear? Okay, you want me to explain the redox lever? Okay, I'll do it. Okay, for the 11th question, I guess yeah, I got 3D. Okay, any more answers? Right, D is the right answer. Um, like all of you know about the glycolysis cycle here. Okay, presuming that all of you know. Um, in the glycolysis, you can get this glucose 6 phosphate, and the lifetime of glucose 6 phosphate is energy rich compound, but it's not a long term energy storage compound. It is a energy rich compound. Whereas glycogen, as you all know, stores, and then when the body is in need of energy, glycogen is broken down again to glucose, and it is used for energy. Similarly, polyhydroxybutyrate PHB and polyhydroxyalkanoates PHA are also a long-term energy storage compounds, but whereas this glucose 6 phosphate is not a long-term storage compound, but it's an energy rich compound that is used in the reaction. I guess the options are a bit confusing, so I'll wait for a few more minutes. Okay, two responses. Okay, others, are you guys trying or should we discuss the answer now? Okay. Okay, let me show the answers first. So D is the right option as you have all answered. So let's start from the fourth, okay, because it's easy this way. We all know NADH is a coenzyme and it usually shuttles this hydrogen and electron transfer occurs. Okay, and ATP is formed by phosphorylation reaction, obviously, because adenosine is the triphosphate. So the formation is by phosphorylation reaction, whereas it is broken down by dephosphorylation reaction. Free energy is released when electrons move from reductant to an acceptor with from a reductant to an acceptor with a more positive redox potential. Okay, so here you can see this chart. Okay, I think this was the thing that Nidhi Praveen was asking. um, the redox tower. Okay, electrons will spontaneously move from donors on the higher end on the tower to acceptors on the lower end of the towers. You can see they are better electron donating molecules. Okay, so when they are going to donate an electron, it's going to be a spontaneous reaction because they are more negative. So here the potential is more positive. You can see in the E naught value or the potential value that is being increased in this manner. So when there is more negativity, there are going to be presence of high number of electrons or high availability of electrons for transfer. So this reaction this way is going to be spontaneous. Okay, spontaneous for electron donation, and these molecules are better uh, acceptors. So when it is very spontaneous, you know that it can give out energy. Okay, so that is what is given here. Free energy is released when electrons move from reductant to an acceptor with a more positive redox potential. So acceptors have a positive, more positive potential, whereas the reductant or the molecule that is going to give the electron, because this get reduced, this is called a reductant. So the lower part of the tower is going to get reduced. So that is why these are called reductant, because they're going to facilitate the reduction reactions. Okay, so when the electrons is going to move from this part, upper part to the lower part, it is going to be spontaneous. When it is going to be spontaneous, we can expect energy being released here. Okay, so that is what, and that is why it is uh, true. Okay, electrons will spontaneously move from more negative potential to more positive potential. Yeah, that we all know from we have learned in electrochemistry or bioelectrochemistry, anywhere that from a negative potential to a positive potential, electrons can be uh, transferred. Okay, uh, is this tower thing clear now, or is there any more dimension of it that has to be covered? Anyone else has any doubts in this tower? If I have to repeat, you can give a thumbs up so that I'll repeat this tower again. Okay, assuming that it is clear, let me now explain the other part. So here you can see all these are energy rich compounds. Okay, you can see most of them contain phosphate in them. So as I told, this phosphate bond when made requires energy, and when broken it releases energy that drives almost many biochemical reactions like glycolysis, TCA cycle. Anywhere you go, you will require ATP, and there will also be formation of ATP there. Okay, so you can see phosphoenol pyruvate, acetyl CoA, acetyl phosphate. All these are also, apart from ATP and glucose 6 phosphate, all these are also energy rich compounds, but they're not like energy storage compounds mostly. So glycogen and stuff, they are like energy storage compounds where you store, and when you are in need of energy, your cell the cell is going to be using them. Here you can see this reaction where um, this ATP formation or ATP breakdown is taking place. If you go this way, it is formation where you can see adenosine monophosphate. In the last session, I think we studied about this, right? We have adenine here, and we have this uh, adenine and monophosphate group with the nitrogenous base. So that is called adenosine monophosphate, and you can have either different types of phosphorylation reactions are possible. So based on that, you can have the addition of phosphate group that can give ADP and ATP. Okay, one important thing to note here is when ATP is hydrolyzed, okay, you get all of you know that you get one ADP and one inorganic phosphate. Okay, but the energy, coming to the energetics part, energetically there is 7.3 kilocalories per moles of energy released for this breakdown reaction. So when ATP is hydrolyzed to give ADP and inorganic phosphate, there is 7.3 kilocalories per moles of energy released. So with that you can relate to the energetics of the process. Okay, and this part of the slide, as we were talking about that aerobic and anaerobic oxidation, you can see that the energy range in case of aerobic is minus 5 to minus 119 kilojoule per electron equivalent, whereas here it is very low. Uh, it is in minus 3.8 to minus 18.8 kilojoule per electron equivalent. Okay, the difference in the reaction free energies for aerobic and anaerobic and organic and inorganic reaction have great effects on the bacterial yield. This is what I was explaining you with that activated sludge process thing and the anaerobic digestion. Okay, when you're studying environmental microbiology, those two are very important processes. One is in the presence of oxygen, another is in the absence of oxygen. So in the presence of oxygen, when you're going to do ASP, your biomass yield will be higher, um, whereas in AD you will not get much of biomass, but instead few products will be produced in that case. Okay, shall we move on to the next last part? Can we move on? Can I get thumbs up from few of you? Okay, okay, okay. I posted the next question. This is again a very easy question. I guess all of you will answer it within a minute now. right? Just to keep you motivated. Simple questions like this. It's copper, yeah. What are all other micronutrients that you know? Maybe one or two. Yeah, magnesium, manganese, yes, zinc, right? Boron, okay. Magnesium, right? Zinc, yeah, right. So why is sulfur, phosphorus, nitrogen, why are they not micronutrients? Like, why do they fall under the category of macronutrients? Yes, manganese is micronutrient, yes. Okay, present in amino acid, yeah, acceptable. Nucleic acids, yeah. Amino acids, right? Right. Anything apart from proteins, nucleic acids, think about everything that you have learned so far so I can get some different dimensions. Right, ATP is right again, yeah. So you will need phosphorus for those phosphate bonds and for the energy that drives life, right? Right, Ruchira, micronutrients, they are needed in lesser amounts based on the nutritional requirements, yeah. Apart from this structural or functional thing, yeah, we have proteins, nucleic acids, yes. One more place that I'm expecting the answer from you people, like where either of this sulfur, phosphorus, or nitrogen is much needed. I'll give you a few more minutes to think because when you think, you can have much more diversified dimensions of what you're learning, okay? Any important place where either sulfur is abundant or phosphorus is abundant, or like not abundant, but very importantly required in that place. Nitrogen, yeah, amino acids. Without nitrogen, there is no amino acids because amine group is the heart of amino acids, right? Accepted. Sulfur, yeah, you have cysteine, right? Okay, okay, Nidhi Praveen, will wait for others also like, to think and let us know. Okay, that's not a very complex thing, but when I say the answer, you will be like, it's very simple and I waited for so long for this. Okay, right. Let me tell the answer now. Uh, how about your plasma membrane? What is that made of? What is your plasma membrane? Right, there that is a phospholipid bilayer, right? So phosphorus is abundant there. And imagine without the phospholipid bilayer, how the cell would look, how the cell would function. So structurally and functionally, phospholipids are much important for the cell, right? So yes, uh, that is what phosphorus is very important in the case of ATP, in the case of DNA, RNA backbone, in the case of phospholipids. If you can consider, you can also consider uh, like almost even phosphorus is associated with carbohydrates also. Okay. Um, so yes, that's the answer that I was waiting for. You have your phospholipid bilayer, and in the case of eukaryotes, we have eukaryotic microbes also, right? In the case of eukaryotes, you can imagine the phospholipid bilayer being present in almost all the organelles that are membrane bound. Okay, even the membrane of the organelles is also made of phospholipids. So imagine in the case of eukaryotes, these are much more important. Okay, so you should have a basic understanding of the concept in a wholesome manner. In this discussion, we are going topic by topic, but at the end of the day, all of this are interconnected. Like it's about a cell. A study is all about studying how its structure is, how, what is its composition, starting from biochemistry, what it functions. You study molecular biology, like how it survives, and this microbial energetics part tell you like microbial metabolism and energetics mainly tells you what reactions or functions they do perform. So uh, microbes generally don't produce products for us, right? When it is going to utilize something or going to produce something, like we may commercialize lactic acid, acetic acid and stuff, but yeah, they have a particular need or particular uh, cause behind whatever they are doing metabolically or biochemically. Okay, so all of this is interrelated, like the biochemical pathways that we generally study with related to organisms. So try to learn all the concepts and see how they are interrelated with each other. That will be very useful for you to understand the concepts also, remember them. So studying with association, um, you can answer the questions in a more easy or uh, more matured manner. Okay, so having said that, I would like to tell you that from next session onwards, I'll also incorporate few questions that will help you think this way, because I feel like when going in a manner where I myself segregate the questions in terms of what topic they fall into, you people start thinking in a way that is very narrow. Like you tend to judge, okay, she's asking from energetics, I have to think from energetics perspective. Okay, so that's not the case. So try reading more and understanding the concepts in a more wholesome way. Okay, now we'll go to the next question. As far as this question is concerned, you have to critically analyze what is a carbon source and what is energy source. If you are able to clearly understand what a carbon source means and energy source means, this question is easy. Okay, I've gotten three responses. I'll wait for two, three more people because the options might be a bit confusing. Okay, let's see the answer for this question. Right. Carbon dioxide is not the energy source for chemoautotrophs. So the discussion for this, I'm going to tell you as such, it'll be a bit confusing for you. So let's see this. Coming on to the table here, this is the periodic table, but with regards to microbial nutrition. So as you can see, we have essential uh, for all microorganisms highlighted in yellow like hydrogen, carbon, nitrogen, oxygen, phosphorus, sulfur, and selenium. Okay, this is what we have seen uh, in that question, that we had nitrogen, phosphorus, sulfur, and copper, right? So copper is, oh, okay. Wait, let me just move through that slide. Okay, so here you can see this is the periodic table um, for microbial nutrition. Here you can see that the elements that are highlighted in yellow are essential for all microbial organisms. So we all know that carbon, hydrogen, nitrogen, and oxygen form the bases, and we have seen why phosphorus and sulfur are important. Selenium is also an essential nutrient for microorganisms. Whereas you can see all these green color things are trace metals, that is, they are required and they have very key roles, but they are required in very small quantity, other trace quantity. So they are known as trace metals. And you can also see few things mentioned as unessential that can be metabolized and that may not be metabolized also. So these are not generally essential for the biochemical reactions to uh, take place. Okay, you can see magnesium, sodium, calcium, all in the place of essential cations and anions, because we all know Mg2 plus can catalyze many enzymatic reactions. Similarly, this ion transport, okay, sodium ion channel, calcium ion channel are all very important. The sodium and chloride, they help to maintain the osmolytic balance of the cell. So they are essential in their ionic form. So the yellow color highlighted things are essential elements, so they are incorporated into compounds or something like that. But when you come to this thing highlighted in this pink color, they are essential cations and anions. Okay, so this is the periodic table which you can um, just have a look into. Coming to the part where this nutrition is being taken from, you can have two energy sources. The energy sources can be chemical or they can be light. When you're So let me first define what energy source and carbon source mean. Energy source means, like, in order to drive your metabolic reactions, you will need energy, like ATP will be there, and you will need energy. So the thing that produces that energy that drives your biochemical reaction is known as an energy source. Whereas for example, in the case of carbohydrate or amino acids, you have this C double bond O something, you when you take glucose, it is C6H12O6, right? So similarly, when there is carbon present in making up the compound or making up the molecule, that becomes a carbon source. So you will essentially need a carbon source, because we know almost all the organic molecules are containing carbon. So carbon source is very very essential to build up any molecule, any biomolecule in our cell, be it carbohydrates, proteins, lipids, or nucleic acids or enzymes for that matter, you will require a carbon source. Okay, so that is the basic difference between a energy source and a carbon source. For example, when you take the energy source can be either chemical or light. When it is going to be light, as we all know, we are going to call it as phototropes. Photo means light, and they are going to derive their energy from light by harvesting the light energy. So they are known as phototropes. We all know they will have some pigment like chlorophyll or something that drives the energy sequestration and hence the biochemical reaction that follows. Whereas when you take in the case of chemical, you will have some um, chemical compound, and when you are going to break down that compound, energy is going to be released, or that compound is going to produce energy by undergoing some reactions, and that energy will drive your other biochemical reactions. So this is what is the definition of energy source and their classification into chemotropes and phototropes. So in the case of chemotropes, their carbon source can either be organic or inorganic. Okay, so when they are going to be organic compounds, you call them as chemoheterotropes. When it is going to be carbon dioxide, you're going to call them chemoautotropes, because when it is going, like, us you take we take our organic compounds as a food, so we are known as heterotrophic organism, right? So in autotrophs, what happens is they take in the carbon dioxide, they fix it into either glucose or something, and then they undergo the glycolysis, and their biochemistry functions that way. So that is known as chemoautotrophs. So they the organisms which have their energy source as a chemical compound and a carbon source is an organic compound are called chemoheterotrophs. And when the energy source is a chemical compound and the carbon source is carbon dioxide, you call it chemoautotrophs. The examples of chemoautotrophs include uh, uh, hydrogen, sulfur, iron, nitrogen, and carbon monoxide oxidizing bacteria. So they are going to oxidize these compounds, utilize carbon dioxide as carbon source, and energy derived from uh, chemical compounds for their biochemical reactions. So in the case of chemoheterotrophs, the final electron acceptor can either be oxygen, it, or it may not be oxygen. In the case of oxygen, almost all fungi, protozoa, and bacteria have their final terminal electron acceptor as oxygen. Whereas uh, there are two possibilities in the case of the final electron acceptor not being an oxygen. Okay, it can either be an organic compound or an inorganic compound. So if it is an organic compound, it can be fermentative. Okay, for example, streptococcus. So when it's going to be fermentative, it is going to produce either lactic acid or acetic acid at the end of the fermentation. Whereas in the case of inorganic compounds, if the final electron acceptor is going to be there, it will again have a electron transport chain maybe. And clostridium is one example. You need not like remember all these examples, just remember uh, what this flow means, like the energy and the carbon source flow means. Okay, similarly when you come to this part where organisms that utilize light, they directly harvest light for their as their energy source, they are known as phototrophs. And based on their carbon source, again the carbon source can be organic or carbon dioxide. Then it is going to be carbon dioxide is going to be photoautotrophs. They are going to be autotrophic. They are going to make their own food, and they have used light for the energy source, so they are known as photoautotrophs. If they are going to use water to reduce carbon dioxide, they will be doing an oxygenic photosynthesis. Whereas if they are not going to use water, they are going to be it is going to be anoxygenic photosynthetic bacteria. Okay, the examples of oxygenic photosynthesis, you can see plants, algae, and cyanobacteria or blue green algae. Okay, that was the question that was asked there. And anoxygenic photosynthetic bacteria include green and purple bacteria, because green and purple as the name suggests, they will have all the colored bacteria have these pigments that helps them harvest light as their energy source. Okay, and coming to this photoautotrophs that use organic compounds as their carbon source, they are known as photoheterotrophs. So the example of photoheterotrophs include green non-sulfur bacteria and purple non-sulfur bacteria. Again, these are also colored bacteria, but they utilize organic compounds as their carbon source. So they are known as photoheterotrophs. I guess this concept of chemotrophs and phototrophs, autotrophs and heterotrophs and all the combinations are clear, right? Can I get a thumbs up or response? Or okay, if there is any doubt, you can either unmute or you can just put it in the chat box. I just saw some hand raised. Is there any doubt? I saw someone raising your hands. Okay, okay. So I'll pose the last question for the day. In the previous concept, be clear what a autotroph or heterotroph is called, or a chemotroph or phototroph is called. Okay, there is no mismatch or confusion in that. Just have that chart clear in your minds, and you can try answering this last question for the day. Right, Parul. I have received an answer from Parul Chaudhary. Okay. I'll just wait for some 2, 3 more people to answer. Right. Okay, a handful of people have answered it right. Yeah, it is. Okay. Glycolysis, anaerobic reactions, and substrate level phosphorylation attributes to fermentation. Why not proton motor force and oxidative phosphorylation? What do they attribute to then? If not fermentation, it's a very obvious answer, but still, I would like to hear from you. Krebs cycle, yeah. Right, Krebs cycle is right, but what's the process in the context of this question? Okay, let me tell you. It's about respiration. Okay. So when it's going to do an oxidative phosphorylation, it's going to attribute to respiration. Whereas when it is substrate level phosphorylation, it's going to attribute to fermentation. And generally, respiration occurs in aerobic conditions. Uh, the one thing that is common to both fermentation and respiration is this glycolysis reaction. So first, the glycolysis occurs in both fermentation as well as in respiration. After this glycolysis process, like you have an organic compound, basically glucose, this undergoes glycolysis and you get pyruvic acid and ATP. Just don't worry about the number of ATPs that is coming here in this process. That's a whole different dimension of energetics here. So again, After the glycolysis has happened, if the glycolysis happens in an oxygen rich environment, it's going to be respiration as told by you. um, it will go into the Krebs cycle and the electron transport chain. If there is no oxygen, there is going to be fermentation, and you either produce lactic acid or ethanol, like, or many other products that will be coming from the fermentation thing. So here I can explain you about this cycle. You need not worry about all the details in this cycle. Just one common thing for both respiration and fermentation is this step. Okay, this step is common for both. First of all, glucose will have its conversion to pyruvate, and you can see there is a generation of ATP in this process. Also ATP is used in this process. You can see ATP is utilized here, here and all, and here you can see ATP being released in this process. Okay, so this pretty much sums up why we are learning about the energetics, why we learned about the energy rich compounds and stuff. Okay, so here you can see this coenzyme NADH that catalyzes the uh, reaction here, and this also helps in energy generation. Okay, so once this glycolysis is done in respiration, what happens is you uh, go to another step that is called as a Krebs cycle or the TCA cycle or the tricarboxylic acid cycle. So after this only this electron transport chain comes. The reason for which I'm showing this is not for all the details, just because of this one word called the proton motive force. Okay, the proton motive force is something that will come into play here. Uh, in this, if you see, there is going to be transport of molecules in, out, as you can imagine, for whatever is being shown here. Let me just make it a bit bigger. Yeah, for what you can see here, you can see protons here, right? So when there is going to be more protons on one side, there is no need of worrying about all the technical details of this process. Just with a normal understanding, you can know that when there is going to be more protons here and less protons on the other side, the protons, there should be a transfer of protons. Okay, obviously there is a concentration gradient, as I told you that a concentration gradient means there will be more molecules on one side and less on other side, right? So here you can see there are more protons here and less. So uh, this will automatically create something called a proton motive force. So this is the entire machinery that helps the mitochondria in doing this electron transport chain and bringing about this production of ATP. As you can see in this part of the diagram here, ATP is produced. So this difference in proton concentration outside and inside the mitochondrial membrane or the mitochondrial inner membrane is known as is what is called as a proton motive force. Okay, so just to explain what a proton motive force is, I have put in this diagram. Other than that, you can just know what glycolysis is and what these steps are. Like, no need of the details of all the process unless and otherwise you are interested to know it. So yes, that was the last question and the last discussion for the day.